Problems with antimicrobial resistance, an overview. Almost as soon as antibiotics were introduced into clinical practice, bacteria were isolated that were resistant to these drugs. Bacteria have evolved numerous strategies for resisting the action of antibiotics and antibacterial agents. This is particularly true of those bacteria that are antibiotic producers. Bacteria that produce antibiotics do so to gain a selective advantage over other competing microbes in their natural environment. If they were sensitive to their own metabolic products, such a selective advantage would be lost. In many hospital units, exploitation of antibiotics is very intensive, and this generates an enormous selective pressure for bacteria to acquire a means by which they may become antibiotic resistant. Under such circumstances, it is not unusual to find bacteria exhibit resistance to more than one group of antibiotics. Resistance to a particular agent may be accomplished by more than one resistance mechanism. Bacteria display antibiotic resistance by one of more of the following. They may lack a target for the antibiotic. Chlamydia do not have peptidoglycan and are not susceptible to the action of penicillins. The antibiotic target may be inaccessible. Peptidoglycan in gram-negative bacteria is inaccessible to penicillins that cannot penetrate the gram-negative outer membrane. Efflux pumps can actively pump out antibiotics from the cell. Gram-negative bacteria resist the activity of tetracyclines by this important mechanism. The antibiotic target may be modified to prevent the action of the drug. Trimethoprim resistance is manifest by alterations in the dihydrofolate reductase target enzyme. Quinolone resistance is affected by point mutations in the DNA gyrase, which prevents the binding of the drug to its target. Antibiotics may be chemically modified or destroyed. Important examples include the huge range of beta-lactamases and the various aminoglycoside modifying enzymes. Chloramphenicol resistance is most often manifest by acetylation by the chloramphenicol acetyltransferase enzyme. Bacteria may elaborate alternative pathways, avoiding the drug target. Methicillin resistance in methicillin-resistant Staphylococcus aureus results from the production of an additional penicillin-binding protein, PBP2', which is not susceptible to inhibition by penicillins. Now let's take a detailed consideration of the mechanisms by which bacteria may resist antibiotics, going back to bacteria that lack a target for the antibiotics. Not all bacteria have peptidoglycan in their cell wall. Rickettsias and chlamydia, for example, lack peptidoglycan. Such bacteria are intrinsically resistant to the action of cell wall inhibitors such as the penicillins and cephalosporins. Although chlamydia do not make peptidoglycan, they do possess penicillin-binding proteins. This raises the fascinating question, why? What role do these enzymes fulfill in a bacterium that does not have a cell wall? Having a target that is inaccessible to antibiotics may be achieved in a variety of ways. The outer membrane of gram-negative bacteria may act as a permeability barrier for antibiotics. Many gram-negative bacteria are intrinsically resistant to antibiotics like benzyl penicillin because such drugs cannot penetrate the outer membrane and so cannot reach their target. Alterations to the side chain attached to the penicillin nucleus may overcome the problem of membrane penetration, and semi-synthetic penicillins, such as ampicillin, have a broad spectrum of activity, encompassing both gram-positive and gram-negative bacteria. Another way in which bacteria deny access of an antibiotic to its target is actively to pump the drug out of the cell. Gram-negative bacteria may resist the activity of tetracyclines through an energy-dependent active efflux of the drug. The antibiotic target may be modified to prevent the action of the drug. Modification of the antibiotic target is often seen in laboratory-generated mutants. For example, bacteria resistant to trimethoprim produce an alternative dihydrofolate reductase. Resistance to the quinolone antimicrobials results from point mutation in the gene encoding DNA gyrase. Aminoglycoside resistance may result from modifications of the ribosome structure. Indeed, in the laboratory, ribosomes may be further altered so that they only function in the presence of aminoglycosides. The drug acts to stabilize the functional ribosome in these aminoglycoside-dependent bacteria. The antibiotic may be chemically modified or destroyed. Many clinically important bacteria produce enzymes that are capable of chemically modifying or destroying antibiotics. Chloramphenicol may be acetylated by the action of chloramphenicol acetyl transferases. Aminoglycosides may be acetylated by aminoglycoside acetyl transferases, phosphorylated by aminoglycoside phosphotransferases, or conjugated with nucleotides, and such modifications render the antibiotics inactive. Antibiotics may also be enzymatically degraded to an inactive form. The beta-lactam bond can be hydrolyzed by a large family of enzymes known as the beta-lactamases. Some beta-lactamases have a preferential activity against penicillins, and these are referred to as the penicillinases. Cephalosporinases are more active against cephalosporins. Recently, broad-spectrum beta-lactamases have evolved that have activity against both penicillins and cephalosporins. There are families of such enzymes that have arisen as the result of point mutations accumulating in the genes that code for the penicillinases. Many of these new enzymes are encoded by self-transmissible plasmids, and these new resistance determinants can spread with very great ease. Bacteria may elaborate alternative pathways avoiding the drug target. Staphylococci have been associated with the production of beta-lactamase for many years. Early in the development of the semi-synthetic penicillins, compounds were manufactured that were able to resist the activity of staphylococcal penicillinases. These drugs had side chains that prevented the staphylococcal beta-lactamase from binding to the antibiotic and hydrolyzing it. Methicillin is a penicillin that is stable in the presence of staphylococcal beta-lactamases and was introduced into clinical practice during the 1960s. Until recently, methicillin was known as methicillin. Shortly after the introduction of methicillin into medical practice, resistant strains of Staphylococcus aureus were isolated from hospital units where the drug was in regular use. Methicillin resistance is greater at 30 degrees than at 37 degrees. Resistance is due to the temperature-sensitive production of an extra penicillin binding protein, pp2', that is not susceptible to inhibition by methicillin. Methicillin-resistant Staphylococcus aureus also produce a beta-lactamase, and they are generally resistant to a very wide range of antimicrobials. Infections caused by methicillin-resistant Staph aureus can thus be difficult to treat. In some cases, the only drug available effectively to treat infections caused by methicillin-resistant Staph aureus are the glycopeptides such as vancomycin. Bacterial resistance to this agent was unknown until recently. Vancomycin resistance first appeared in the enterococci, and these bacteria are resistant to all currently available standard antimicrobial therapies. In an experiment of dubious ethical status, the gene encoding vancomycin resistance was transferred in the laboratory from a vancomycin-resistant enterococcus into an MRSA. New antibiotics are now available to treat MRSA infections. The mobility of antibiotic resistance determinants. Some antimicrobial resistance genes have only ever been found located on the bacterial chromosome. Others have been found to lie on plasmids. Plasmids encoding antibiotic resistance are often referred to as resistance factors, R factors, or R plasmids. R plasmids may encode resistance to several unrelated antibiotics. Some R plasmids are self-transmissible and can move from strain to strain, even between different bacterial genera. Other R plasmids, although not self-transmissible, may be mobilized by other plasmids, and they need not necessarily encode antibiotic resistance. Antibiotic resistance genes are frequently located within transposons. These can move more or less at random around a bacterial genome. Genes encoding antibiotic resistance are frequently located within transposons. These can move more or less at random around the bacterial genome. Genes encoded by transposons may thus spread very easily, because many transposable elements may become associated with transmissible plasmids. In this way, antibiotic resistance genes may become rapidly disseminated. Control of antimicrobial resistance in pathogenic microbes is one of the greatest challenges currently facing medical microbiology. If we are unsuccessful, we will surely enter the post-antibiotic era.